nuit. Dors, la nuit est noire, dors, mamie.
Okay, welcome everyone. Here we are again. Today we are going to talk about biology. The topic is sleep universal? Our tonight's speaker is Pierre, and as a curiosity, Pierre studies entomology in National Taiwan University. He is doing a research on cockroaches and sleep. But today he's going to talk about sleep across the whole evolution, across the whole Earth. So, Pierre, you can start.
Thank you. So, first of all, thanks for having me here. Usually when you meet people in Taiwan, they ask you what you're doing. So I say I'm a student. Then they ask me, you're a Chinese student? I say, no, I study entomology. And they say, oh, anthropology, humans and culture, that's good. I say, no, the study of insects. And then they might ask, what do you study specifically? And I say, the sleep of the cockroach. So that's usually when they burst in laugh. And so I hope with this topic I can make you guys understand what's the framework of my research of the sleep in the German cockroach. And this study might help us to respond, to answer this question, is sleep universal, and what's the core function of sleep? So I first give you the outline, so I do a brief introduction, then I talk about sleep in mammals, then sleep in other animals, and finally a discussion part. And there's also a number at the top right corner of the PowerPoint, so if you want me in the discussion to go back to the slide, you can just note the number. So, I would like to start with the famous quote from looks like a Russian biologist, that nothing in biology makes sense except in the light of evolution. Indeed, sometimes we have biological behaviors we don't understand, and it's very good to look that behavior through the looking glass of evolution. And we still don't know why we sleep, and I think looking to sleep through the evolution can help us. And we don't know why we sleep, because while sleeping we cannot forage, we cannot get food, we cannot eat, we cannot mate, like those two beautiful dragonflies, and we cannot protect ourselves. We are very vulnerable. So we still don't understand why really we sleep. And studying sleep phylogenetically might give us an answer. So the phylogenesis is basically the relations between organisms. Like phylogenetic trees, they show relations between different organisms. I'm going to have one later. And the idea is like, here's the eye of different mollusks. And we can see the most complex eye is very complex. And that would be our sleep. So with dreams, with different phases of sleep, it's very complicated. But if we study the eye of simpler mollusks, it's just some simple cells that receive light. And the idea would be that sleep in more ancestral organisms might be simpler, like this eye. And so it could give us a better insight of why we sleep. And so first we should define what is sleep, how to define sleep in an organism. So there's 4 criteria. First one is behavioral quiescence, so basically a reduced motor activity. When we sleep, we lay down, we don't move that much. The second one is elevated arousal threshold, so like we need higher stimulus to wake up. Like we need to shake you so you can wake up. That's the idea. The homeostatic regulation would be that if you don't sleep for a night, the next night you will sleep more. So there's a regulation. And finally, the last one is a species-specific position. So for us, we lay down, we close our eyes, and every animal will have a different species-specific posture. And those 4 criteria look quite simple, but it's actually very hard to prove them for any kind of animal. Okay, so let's talk a little bit about the sleep in mammals. We have a very good criteria to study sleep in mammals. It's the electroencephalogram, EEG. It's basically electrodes that you put on the head of the patient, and it records the overall activity of the neurons of the brain. And we found out in human that we have 2 phases of sleep, 2 different phases. We have what we call the slow wave sleep, with like large amplitude waves and low frequency. And we have the rapid eye movement sleep with like very short and high frequency waves. And you might know the REM sleep. It's when we sleep we have so those small rapid eye movement, and that's also the time when we dream. That's when dreaming occurs. Okay, and if we talk about the sleep time within mammals, we can see that very closely related mammals like this cat and this genet, or the two squirrels, they are closely related, but they are very different total time of sleep or rapid eye movement of sleep. So for example, the very lazy owl monkey, he sleeps 17 hours, but it's a monkey, it's quite closely related to us, whereas us humans, we sleep about 8 hours. But conversely, some very distinct animals have very close total sleep time or REM sleep. So for example, let's say the goat and the eastern tree hyrax, they're very different, but they have the total same amount of sleep. So there's some things very weird about sleep, even in mammals that we kind of know very well. And what about marine mammals? Oh, no, sorry, not yet. Another difference would be the arousal threshold, so when it's the most difficult to wake us up. So for the rat, for example, it's during the rapid eye movement, so during the dreaming phase. It's the hardest period to wake them up. And for humans, it's the opposite. Okay, so there's weird differences. What about mammals? We have marine mammals. We have some marine mammals that basically move all their lives because they have to, either for breathing and stuff like that. So that's also a problem, because I said that one of the criteria to define sleep was the reduced activity or stopped activity. But those mammals, they keep moving all the time. And what about the dolphin? Dolphins, they found a way to counteract that. They sleep with only one part of their brain, so the other half of their body is active, and then they switch hemisphere, so they can rest one hemisphere and still move, and then they switch to the other hemisphere and are still able to move. So there's a lot of diversity in sleep traits in mammals, and the idea is to study sleep with a comparative phylogenetic approach. So to study sleep in most animals we can, and animals very different, like cockroach, birds, worms, and things like that. And usually cockroaches, those more ancestral animals, they are easier to study, especially the molecular and genetic mechanisms. Okay. I'll talk a little bit about sleep in some model organisms. So in biology, model organisms are organisms that a lot of biologists study. So we have the zebrafish, we have the Cenorhabditis elegans, which is a flatworm, very small, just a few millimeters, and the famous Drosophila melanogaster, which is the fruit fly, the common name. So the zebrafish, for example, advantages. Conserve neurotransmitters and neuroanatomy, because they have a cord, they have a backbone like us. That's pretty useful. And it's been shown that they show all the criteria of behavioral sleep, so the 4 criteria I introduced earlier. What about C. Cenorhabditis elegans, this very small flatworm? It's a very interesting animal because every single individual has always the same number of cells, which is 959. It never varies, always the same number of cells. So it's a pretty useful tool. And it's been shown that there is a sleep-like state. And if that flatworm presents a sleep-like state, it's probable that the common ancestor of all bilaterally symmetric organisms may show sleep. So bilateral organisms are organisms with a left-right symmetry. And before that, it's the radial symmetry, so circle symmetry basically, and that would be the jellyfish or sponges. So to answer the first question, is sleep universal? Probably for organisms that show a bilateral symmetry. but for jellyfish and such, we are not sure. So that's an element of answer. So yes, about this flatworm, they sleep about 2 or 4 hours every 10 or 16 hours. So it's not a 24 hours cycle. And they have a reduction in responsiveness, so the arousal threshold. It's harder to kind of make them move when they're in that state. And when you deprive those worms of that sleep, the next day they will sleep more. So once again, those criteria. And what about the species-specific posture? Just about a year ago, was shown that when they sleep, they have this more round body posture, and the muscles, the tones of the muscles are reduced too. So here is when they are active, and here would be the species-specific posture of the sleeping flatworm. Okay, so this worm, yeah, as you see, the scale, this is 190 micrometers. So it's what? It's 2 millimeters or something? Even less. Very small. Okay, so that's the superstar of biological organisms. We know a lot about Drosophila melanogaster. So of course, a lot of studies on it has been done about sleep. And what we have a very powerful tool. That's what I use in my lab, partly with my cockroaches. It's a tube with a laser at the middle, and every time the fly crosses the laser, it records 1 beam. So then we can have actograms. So it shows the activity on 2 weeks. We can have, so it's very simple and powerful tool to study the sleep of the Drosophila melanogaster. This we don't need. Now I'm going to show the cuttlefish. It's been shown that the cuttlefish also has a sleep behavior, and moreover, it's been shown that they have 2 phases of sleep, like us, like slow wave sleep and rapid eye movement sleep. So maybe cuttlefish might be dreaming, and I'm going to show you the video. It's pretty interesting. So maybe a bit wrong. So here's the 2 eyes. Here you can see the pointer. And here's the animal. And you will see that when it enters that second phase of sleep, it will activate its chromatophore and change color, and not in a random way. It's going to show patterns. And they also twitch their little arms, and they have some movements of their eyes. So it should be coming soon. Maybe let's fast forward. Okay. It's coming. So animal is here. Up. So it's not random, as you can see. It's kind of, it shows patterns. We can't see the eyes really, but they show some twitching movements too. And that was a recent paper too, maybe a year and a half ago. And it was the first time they showed 2 phases of sleep in animals other than birds or mammals, invertebrates. So we don't really know why it shows that second phase of sleep, but it has probably significance, but we don't know why. So that was the dreaming cuttlefish. What next? So sleeping insects. You probably know that arthropods, there's a huge number of arthropods, of species, compared to vertebrates or other animals. And the problem is, this would be this part here is the representation in studies addressing sleep duration. So there's kind of a problem. Like, there's a huge number of insects, but like very, very small studies concerning their sleep. Whereas, as you can see, mammals and birds are very well studied. But they show small numbers of species overall. So I think that's why sleeping insects could be interesting. So that is a phylogenetic tree. It's a bit small, I'm sorry, but it shows basically the relations of all group of insects. And so insects, they are very diverse. But concerning the sleep, only 2 groups have been studied. The Hymenoptera, that's the bee. The bee has been studied a lot. And, the Diptera. Diptera is the fruit fly, Drosophila melanogaster. So all the other groups, we don't know about them. And can I find the cockroach somewhere here? Yes, Blatody. This is the group I'm going to study. And so far, I think I'm a pioneer for Blatody and sleep. That's pretty cool. So, how to define sleep in insects? Once again, we have to define to prove those 4 criteria: behavioral quiescence, species-specific posture, increased arousal threshold, and homeostatic control. So I'm going to show you here how to do that basically, and a few figures. I hope they're going to be big enough on that screen. So, to study the behavioral quiescence, it's to study basically if it stops moving at some times of the day. So here we can see, not very clearly, but there's a lot of activity here. Here's the activity, and here's the time. And during the night, flies stop moving. Okay? And it follows the circadian time, it follows the day. Every night they stop moving, and during the day they're active. And this is using the tube I showed you. So basically this is like they cross a lot, the laser, and during the night they stop moving, they just rest somewhere, and they don't cross the beam, so that's why we have this. Here's just a zoom part, it's not important. And this is for my lovely cockroaches. It's an actogram. So every dot represents activity, same when they cross the laser of the tube. And here you can see the white bar is with daylight, and here all this part is nighttime. So you know cockroaches are nocturnal animals, so most of their activity is during the nighttime. You can ask me about that later. Anyway, the species-specific posture. So for that you just have to film them and see if there's a posture. For the fruit fly, they usually stay near the food. The food is at one end of the tube. And you can see in about 5 minutes, they start slowly to get closer to the floor of the tube. So that's the species-specific posture. For my cockroaches, that's what I got from my recording. This would be the active position. When they're moving, they're kind of high on the legs and with active antennae. And if they sleep, they might have, that would be the sleeping position. So similarly with the Drosophila, they get closer to the tube of the floor of the tube. And we can't really see here because the picture is cut, but the antennae drops, they become parallel to the floor, whereas usually they are kind of, there's an angle. Okay, so that's the species-specific posture. What about the increased arousal threshold? So that figure basically shows, here is the intensity of the stimulus, and here they use a mechanical stimulus. So basically you have to tap the tube. And you can see that during the night time, so when they sleep, you need much more intensity of stimulus or number of stimulus. Okay? Whereas during the daytime, they are active, so you just tap them up, they become active. But if they sleep, when they sleep, you need stronger stimuli to activate them. And finally, the homeostatic control. That's the, I told you, you don't sleep a night, next time you sleep more. So basically you have to do that with the insects. So you deprive them of sleep or rest for one night, and you just check the next night if they rest more, if they sleep more. I'm going to show you a cool study they did on bees. They built what is called the insominator. So it's basically a box when you can deprive the bees of sleep. So at the center of the frame you have the hive, and here you have a magnet and a magnet array. Okay, so you have magnets that they can sweep the hive like this. And what they do next, they put on the bee some small magnetic piece of metal, and they mark the bee. So they just put some paint so they are able to recognize them. And so those bees, that some have that metallic tag, some don't. So when you swipe the array, the magnet, the bees are going to be jogged, they're going to be shot like blue because of the magnet. So that's a way to deprive them of sleep. And then after so for one night they sweep the array quite regularly, so they deprive the bees of sleep, and then they just check if they sleep more the next night. And that's this figure. So the treatment bees, the dark the darker curve is the one that have been jolted, the one with the piece of metal. So you can see that they have more slip, right? This is the slip here. Whereas the control bees, so the bees that didn't have the magnetic tag, they don't show significant higher time of sleep. Yeah, makes sense. Okay. And yeah, this study was made to see if the dance of the bees was affected by sleep. Okay. So maybe you heard about the waggle dance of honeybees. It's a way for them to show where's the food to their mates, to the other bees in the society, basically. And so the dance has two components. First is the angle with the vertical. The angle shows the direction from the hive. It's the angle from the hive to the food source. Okay. And then you have the duration of the waggle, the duration of the waggle dance. This indicates the distance. So if you have the angle from the hive and the distance, basically you can find the food source, right? And so what they did, they deprived some bees of sleep, as I just showed, and then they checked the precision of that dance compared to other bees. So first I show you the waggle dance, what it looks like in real. Ah, this one is shuffling. One more up. So the duration of the dance is the distance once again, and the angle between to the vertical is the angle from the hive to the
It's very
source.
fast.
Yeah, and somehow they can, the other bees can recognize. I mean, they sense it very well and can find the food source very easily.
Put some music.
Can you put some music here? Okay, I have another one quite similar, I think. Maybe it's clearer. Or... No, it's not. But you get the point of the waggle dance. Every day they're shuffling. And so then they check the precision of the waggle dance. So here is before the sleep deprivation, okay? And this is after post-night disturbance. And the cross points here are the bees that were deprived of sleep. And you can see this is the error margin between where's the food source and the angle. And you can see that the bees that didn't sleep do much more error compared to the bees that sleep. Bees that slept, they are pretty accurate, but these ones are not. And this is post-night, after the doesn't matter. So basically, sleep is important for societies, because if all the bees in the society, they live in societies, can't sleep, they won't be able to find the food as good as controlled societies. And I think it's time, no?
6 minutes, 7 minutes.
Left? The time. Okay. I'm just going to finish by my further work I want to do. I first want to define the sleep of the cockroach. I'm still working on it, and it takes a long time. But then I would like to study the sleep duration and the link between molting. Cockroaches, they molt, so they go from small nymphs and they molt 5 or 6 times before becoming adults. And this is not the case of bees or the fruit fly. The bees and the fruit fly, they just from larvae, they become adults directly. That's not the case of the cockroach. And there might be links between molting and sleep duration. So I would like to study that. Another thing that's the sleep duration at different instars. Like, for example, we know babies, they sleep a lot because they need their brain to develop. We know that sleep is linked to brain development. And so I would like to see maybe younger nymphs of cockroaches, they probably sleep more than adults. So that's the point. And investigate molecular mechanisms underlying sleep. So it looks difficult, but basically it's providing caffeine, basically, to the cockroach. Put some caffeine in the food of the cockroach and see if they sleep less, or they should sleep less. And so that would show like there's conserved mechanisms, because when we drink caffeine, when we have caffeine in our organism, we sleep less. So there would be like common pathways underlying sleep between mammals, us, and cockroaches. The fruit fly, if you provide the fruit fly with caffeine, they sleep less. That's known already. And also, that would be quite hard to do, but to investigate sleep and memory. We know that sleep is very important for memory, but we're not sure if it's true for cockroaches or other animals, so I would like to study that too. The problem is it's very hard to train cockroaches. The idea is to train them to do something and to test them again the next day with some cockroaches that didn't sleep and some that did. With the bee, it's very easy to train the bee. We have many methods to train bees to do stuff, so it's kind of easy with the bee. But with the cockroach, they're kind of stubborn. It's hard to train them. And now that's it. Thank you.
All right, Pierre, thank you.
Dors, mamie, c'est notre dernier soir. Dors, mamie, je pars.